I'm like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hello and welcome to the National University of Singapore Yong Lu Lin School of Medicine Healthy Longevity Webinar. Thanks for joining us again tonight. We have a great talk from Ray Mak in a few minutes. I also want to just point out that we've had two exciting things happen, I think, in the aging field in the last week. One is the first International Gerophysics Conference, where we brought together the theoretical physicists with the aging biologists and tried to see if we could think about how to describe aging from a mathematical set of equations. Early days, but I think there's an interesting area to explore there. More to come. Also, I was just at the RISE Longevity Conference in Bangalore, India, and I was really excited to see how much focus India is starting to put on the aging process and understanding what's similar and unique in the Indian aging population. So another country joins the fray, so that's exciting. Before we get to the main talk tonight, I want to introduce Dong Han, who's a research fellow in Yin Tsai's lab, and he will present on his own study, limiting cap-dependent translation increases 20S proteasomal degradation and protects the proteomic integrity in autophagy-deficient skeletal muscle. Big title.
Thank you for introducing. Hello, everyone. As mentioned, my name is Dong Han from Dr. Cai Xin's lab. Today, I'd like to share with you about my study about the role of 4-EBP1 in regulating protein degradation. As for the introduction, I'd like to start from the term called sarcopenia, which means an age-related loss of muscle mass and muscle function. The underlying mechanism of sarcopenia included the muscle mitochondrial dysfunction, muscle satellite cell dysfunction, increased muscle inflammation, and most importantly, the imbalance of protein homeostasis in skeletal muscle. The key regulator of protein homeostasis is mTORC1, which can regulate both protein synthesis and protein degradation. Hyperactivation of mTORC1 has been shown to cause severe myopathy due to the defect of autophagy. Our group's previous publication shows that activation of mTORC1 downstream cap-dependent translation inhibitor, 4-EBP1, can rescue the phenotypes observed in mTORC1 hyperactivated mice. In my project, I'm also going to focus on 4-EBP1 and try to understand the underlying mechanism how 4-EBP1 can rescue the protein degradation systems. To dissect the underlying mechanisms, we use an autophagy-deficient mice model in which a key regulator of autophagus maturation, ATG7, is knocked out specifically in skeletal muscle. We also generate another mice model to activate 4-EBP1 in the skeletal muscle of ATG7 muscle knockout mice. Our result shows that when we knock out ATG7 in skeletal muscle, it will cause severe protein aggregation as shown by accumulation of ubiquitinated protein and the autophagy adapter P62, as well as LC31, which is a precursor of autophagous membrane marker LC32. However, these phenotypes can be rescued by activation of 4-EBP1. But when we activate 4-EBP1, we do not see any increase of LC32, which suggests that this relief of protein aggregation is not through autophagy. So we start to look at the ubiquitin proteasome system, which is another major protein degradation system in the eukaryotic cells. In eukaryotic cells, we mainly have three different types of the proteasomes. the constitutive 26S proteasomes, which require ATP for its proper function, and the constitutive 20S proteasome and immunoproteasome, which can work in an ATP-independent manner. We first use an ex vivo assay to measure the ATP-dependent proteasome activity. Knockout of HG7 increases the ATP-dependent proteasome activity, which might be a compensate to the autophagy defect. Activation of 4-EBP1 shows further increase. However, our ex vivo assay is work in an artificial conditions, which we provide sufficient ATP for the proper function of the proteasome. Whether this conclusion in the ex vivo study can be applied into in vivo condition, to confirm this, we use the staining to measure the muscle fiber proportion in the skeletal muscle of our different transgenic mice. As we can see from the staining result, the first row is the extensor digitorum longus muscle, EDL, and the second row is the soleus muscle, SM, and each column refer to a different transgenic mice model. As you can see from the most right column, the 4-EBP1 mutant HG7 muscle knockout mice shows more green fibers compared to the control and HG7 muscle knockout mice in EDL, and it should have more blue fibers in SM compared to the control and HG7 muscle knockout mice, which suggests that activation of 4-EBP1 increases the proportion of oxidative fiber in skeletal muscle. The oxidative fibers are slower twitch and they have more mitochondria compared to the glycolytic fiber. So generally, muscle with 4-EBP1 hyperactivation have more mitochondria, which indicate a higher mitochondrial biogenesis. We also measure the ATP-independent proteasomal degradation capacity, which is also increased by activation of 4-EBP1. Our Western blot results suggest that this increase of ATP-independent proteasomal degradation capacity is mainly through increase of biogenesis of immunoproteasome. Taken together, our results suggest that, firstly, activation of 4-EBP1 can rescue the protein aggregation phenotype in autophagy-deficient muscle by increasing the ubiquitin proteasome system. Secondly, the activation of 4-EBP1 can increase mitochondrial biogenesis to provide energy supplement for 26S proteasome in autophagy-deficient muscle. Last but not least, activation of 4-EBP1 increases the biogenesis of immunoproteasome, which can provide ATP-independent proteasomal degradation capacity. Thank you.
Thank you. The TOR pathway continues to be one of the major pathways for longevity, and trying to understand what's downstream of mTOR, including 4-EBP1, S6 kinase, and other targets, is very likely, I think, to teach us more about the mechanisms of aging. We're going to switch course a little bit, though, and Dr. Raymond Mak is an associate professor of radiation oncology at Harvard Medical School and the Artificial Intelligence in Medicine program at Mass General Brigham. His research focuses on the development and clinical implementation of artificial intelligence, including developing algorithms of imaging biomarkers to predict health status and outcomes in cancer. Tonight, he's going to be talking about clinical application of artificial intelligence to quantify biologic age from face photographs, or FaceAge. Thanks for joining us, Ray.
Thank you for the introduction, Dr. Kennedy. I want to thank you and Dr. Mayer for the invitation to speak here today. And I'm looking forward to sharing how we developed this new AI algorithm that we call FaceAge that utilizes just a simple face photograph to help quantify biological age. These are my disclosures. So I'm a radiation oncologist, and I spend a lot of my time treating lung cancer patients, and I look at the concept of biological aging through perhaps a slightly different lens than the healthy longevity community. In cancer care, we're often using chronological age as a key prognostic factor in many of our risk models and to help us drive clinical decision-making. However, most oncologists will tailor the intensity of cancer treatments based on the functional status or biological age of a patient. You'll hear us say in the clinic phrases like the patient looks older than stated age, or the patient doesn't pass the eyeball test, as we clinicians try to qualitatively assess biological age to inform personalized treatment decisions. And for a cancer patient who may be biologically or physiologically older than their chronological age due to the impacts of lifestyle and environment and genetics, this is the patient who might have to de-intensify treatment for or provide more support to get them through toxic cancer treatments. This disconnect brings to mind Oscar Wilde's story of A Picture of Dorian Gray, where the main character sells his soul for immortal youth, but his portrait bears the evidence of his decay and aging that's brought about by his vices. Conversely, we also encounter the polar opposite situation in the clinic, where an elderly patient, based on their date of birth, may appear biologically and physiologically much younger than their chronological age. And that may be the patient we want to intensify treatment for and treat them as if they are a younger person. So there are many methods being developed to quantify biological age, from blood-based biomarkers, genetic biomarkers, and medical imaging. But perhaps the most intuitive measure is the appearance of a person. We look at someone and we think, wow, they look really tired today, or they don't look well. And as I said, doctors routinely use clinical gestalt or the eyeball test to make these qualitative assessments of a patient's overall health to guide decision-making. Thus, we hypothesized that we could quantify this human and clinical intuition by developing facial health recognition AI that takes in as input a simple face photograph and extracts imaging features to predict a person's biological age. A big part of the motivation behind us starting this research several years ago was that at that time there were advances in AI technologies called deep learning that had really improved the accuracy of facial recognition algorithms. You may have noticed this in your day-to-day life, that the facial recognition applications you use to unlock your smartphone, or applications that automatically tag an individual's face in photographs on social media have really gotten a lot better in the past few years. These gains are driven by these deep learning approaches, which are AI approaches that utilize large training data sets, such as a large face photo database, along with neural networks that have an architecture that mimic how the human brain works, and learns to extract quantitative imaging features from photographs to create a unique identifier for each person. So once you've trained this algorithm and created a database of face photos that includes the quantified facial features, then when you go to use a facial recognition system on your smartphone, for example, you take the face photo and it runs through the deep learning algorithm, which will automatically detect and localize the face in the photograph, extract quantitative features, which are then matched against a database of faces, and returns a identity verification. So we leverage these advances in AI-based facial recognition to test our hypothesis that a person's biological age is quantifiable from facial characteristics captured in a simple face photograph. And thus we trained a facial health recognition AI that we call FaceAge, which is built off the backbone of facial recognition AI in that it takes in a simple face photograph as input, localizes the face, and extracts imaging features. But instead of producing a final output of predicting the identity of a person, we've trained it to output a biological age estimation. So you can see in this example, the algorithm projects that this individual's face age looks 5 years younger than their chronological age, the age based on their date of birth. This work is in press at the Lancet Digital Health Journal, but a preprint is available via the embedded QR code. To give you a sense of how we developed the FaceAge algorithm, we started by training the algorithm on over 50,000 face photos of presumed healthy individuals from a publicly available database, the IMDb Wiki dataset, which includes both face photos and the individual's age at the time the photograph was taken, allowing the algorithm to learn facial features correlated with a given age. We validated performance of the algorithm in accurately predicting age in over 2,500 healthy individuals from a separate public data set called the UTK dataset, which consists of non-famous individuals and it has a wider range of ethnicity. As I mentioned earlier, we trained FaceAge with an eye toward clinical applications in cancer care. And since we discussed doctors routinely use clinical gestalt or the eyeball tests to make qualitative assessments of patient health, we wanted to see if FaceAge could quantify this clinical intuition and be a prognostic biomarker to guide decision-making in cancer care. To address this clinical application, we initially gathered a data set of over 6,000 cancer patients from 2 cancer centers in Europe and Boston. We're fortunate to have a relatively unique data set in medicine to be able to test our clinical application. About 50% of cancer patients will need radiation therapy at some point during their cancer care. As part of our routine clinical workflow, every radiation oncology center takes a digital face photograph of a cancer patient before they start treatment for identification purposes. Our field is highly digitized, so the face photographs, medical imaging, like our CT scans and clinical data, including the patient's age and outcomes, are all stored and linked in our clinical databases. And I want to emphasize that these photos are fairly low quality and that they're taken with a consumer-grade digital camera, with the patient standing in a clinic room in a non-standardized manner, with different lighting conditions. So a selfie from a smartphone would likely be much higher quality than these photographs that we used. So we deployed the FaceAge algorithm on the face photographs of our healthy individual and cancer patient cohorts, and we generated FaceAge estimates for each individual. We observed that cancer patients on average are significantly older appearing than people without cancer. And this plot shows AI predicted face age minus chronological age. On the left, healthy individuals center around a delta of 0, so their age congruent, while cancer patients appear about 5 years older than their chronological age. And this was observed over multiple cancer types, including breast cancer, gastrointestinal, lung, prostate, head and neck, and patients with metastatic cancer in 2 independent cohorts. In our cancer patient cohort, older face age was associated with worse survival. And here's a Kaplan-Meier plot of 4 cohorts with different face age ranges, with individuals who appear older than 85 doing the worst, and patients who looked younger than 65 had the best survival outcomes. On multivariate analyses, older face age remained predictive of worse survival in cancer patients, even after adjusting for chronological age, meaning, again, the age based on the date of birth, gender, and cancer type, and provided additional explanatory power beyond chronological age. We then wanted to test the clinical application of FaceAge and focused on one of the most challenging prognostic tasks in medicine, and that's to estimate survival time at the end of life, which has important implications in cancer care where we don't want to overtreat at the end of life. A classic question that we ask clinicians in this context is whether they would be surprised if a given patient would die in the next 6 months. So we designed an experiment where we asked 8 oncologists and 2 lay people to predict whether a patient would be alive in 6 months. In the first phase of the survey, we asked the end users, but we provided the end users with only the photographs of 100 cancer patients from a palliative care cohort and asked them to predict whether the patient would be alive in 6 months. In phase 2, we gave them the photographs again, but now accompanied by standard clinical information, including key clinical prognostic factors in this care setting, and asked them to answer the question again. Lastly, we then shared with them a FaceAge-based risk prediction for each patient and asked them to answer the 6-month question for a 3rd time. Not surprisingly, our human clinicians did not make very accurate end-of-life predictions by just looking at the photograph of a patient, and there's a wide range of performance. The accuracy of some of the estimates made by our oncologists, as shown by the area under the curve plot here, was just slightly higher than a coin flip. But you can also see that there are a few doctors who are very good at this task. After we gave the doctors some clinical information, their estimates improved, but the performance was still not great. But when we provide the clinicians with FaceAge information, their end-of-life predictions improve significantly. So we believe that FaceAge can help clinicians by providing quantitative data to improve their clinical intuition and support their decision-making at the end of life. In ongoing work, we further validated FaceAge as a clinical prognostic biomarker in an expanded real-world data set of over 24,000 cancer patients that include the most common cancer diagnoses. In this clinical validation study, we focus on extreme phenotypes, the patients with the highest discordance between FaceAge and chronological age. And notably, 13% of patients in this cohort had a FaceAge that was more than 10 years older than their chronological age. We observed that patients with advanced metastatic cancers or very aggressive cancer types, which have the worst prognoses, like aggressive leukemias, pancreatic cancers, lung cancers, were more likely to have a FaceAge minus age difference of greater than 10 years, meaning these patients were the ones that appear much older than their chronological age. In contrast, people with more indolent or less aggressive cancers, like prostate cancer, indolent lymphomas, and a certain kind of less aggressive breast cancer called DCIS, were less likely to have such a discordant chronological age versus face age. They're more age congruent, more like the healthy population that we saw earlier. In this large cohort with long follow-up, we observed that face age minus chronological age difference was significantly associated with survival. And the greater the discordance, the worse the survival. As you can see in this plot on the left, if patients with a face age older than a chronological age by more than 10 years or more than 15 years had the worst survival. Furthermore, younger cancer patients who had large face age minus age differences had prognoses similar to elderly patients. So their biological age trumped their chronological age. And you can see that on the plot on the right. We observed these associations across multiple cancer types and subgroup analyses in multivariate models adjusting for other known prognostic factors, validating face age as a novel prognostic biomarker in cancer care. We also looked at the association between face age and lifestyle habits, including smoking, alcohol, and substance abuse in our cancer patient cohort. Individuals who are current smokers, heavy drinkers, and self-reported substance abuse had a significantly higher face age minus age difference, meaning that they looked older than their actual age. Interestingly, former smokers had a lower face age to age differential, sort of landing between current smokers and never smokers, suggesting a potential reversal in biological aging with quitting smoking. With any AI algorithm, though, we are always concerned about the black box nature and whether the model predictions have underlying biology or physiology that can be interpretable by human clinicians or users. To address that issue, we performed attention mapping, where we identify the areas on each face image that are most important for the algorithm's prediction, and then map these areas to a standardized 3D model of the human face. You can see on the right, the attention map averaged over 3,800 cancer patients, and that the algorithm seems to be relying on features from the periorbital areas around the eyes, the anterior cheek, the nasolabial fold, and the temporalis regions, which is consistent with what regions of the face humans might look at to determine someone's age and what's reported in the facial aging literature. As an exploratory analysis to further understand the biological basis of face age, we also evaluated the association of single nucleotide polymorphisms, or SNPs, in 22 senescence-related genes with face age. And we used a data set of 146 lung cancer patients who'd previously undergone germline whole exome sequencing. We identified an association between elevated face age and the cell cycle regulation gene CDK6, while observing no such association with the chronological age at diagnosis. CDK6 has been implicated in aging and senescence, and we're following up on this analysis with a genome-wide association study in a larger cohort. So where do we go next with this research line? In ongoing work, we're applying face age onto serial photographs obtained longitudinally during a cancer patient's care to understand how changes in facial aging rate may be even more prognostic than a single measure. Additionally, we're utilizing newer AI training approaches called foundation models and self-supervised learning to develop next-generation facial health recognition algorithms. Going a little bit deeper into the technical approach behind our second-generation algorithms, we're utilizing a mass autoencoder in a dataset of about 40 million face photos. The training involves masking random pixels on every image for self-supervised learning, where the algorithm learns on its own from the existing visible patches to reconstruct the missing data. And you can see some examples of how our facial health recognition foundation model performs at this reconstruction task on the right. But more importantly, through this training process and on over 40 million facial images, the model learns a diverse set of facial imaging features. These features can then be used as a foundation for us to train a wider variety of facial health algorithms, including we're currently developing a more accurate second-generation face age algorithm, and then additional algorithms that can actually predict different health states like the likelihood of death or likelihood of a serious illness from face photographs. We certainly recognize, though, that FaceAge and our continued development of new facial health recognition AI will face ethical and technical challenges, which mirror some of the concerns raised with facial recognition technology more broadly. First, we have to ensure that our algorithms' predictions are generalizable to a diverse range of faces from people of different races and age ranges. And we believe that our new foundation model approach will help mitigate some of these concerns. Secondly, we have to demonstrate that the algorithms perform robustly, meaning they generate accurate predictions, despite alterations in conditions like individuals wearing makeup or photos from different lighting conditions, and in people who have undergone cosmetic surgery like facelifts. Lastly, we need to deploy these algorithms in a way that safeguards individual privacy and prevents misuse. Looking to the future, while face photographs represent the most intuitive place for us to deploy AI-based biological age estimation, cancer patients also routinely undergo X-rays, CTs, MRI scans, which contain additional hidden quantitative data on the biological age of a patient. For example, other members of our research group have developed AI algorithms that automatically quantify coronary calcium on CT scans, which is a measure of the biological age of an individual's heart, or estimate the biological age of the brain from staging MRI scans, or an individual's lung age from a chest CT X-ray or a chest CT or a chest X-ray. And the power of these AI-based approaches is that we can opportunistically assess multi-organ biological age through non-invasive serial imaging that's already part of our routine clinical care for cancer patients. While our focus in oncology has been to leverage these multi-organ biological age estimates to automate risk stratification and personalized cancer therapy selection, we can also easily apply these imaging-based age estimations to quantify aging in more healthy adults, such that a given individual might get multiple readouts on the aging rates of different parts of their body, and we can potentially correlate them with other multi-omic measures of biological aging, including genomic, transcriptomic, proteomic, and the microbiome. This is an area where we're starting to explore further and would love to partner with experts in longevity and aging science. So I hope I've shown you that how FaceAge technology can have an impact on prognostication in medicine. But what about the impact for people who aren't sick and don't need medical attention? For people at home, AI-based quantification of biological age from face photographs could also provide a non-invasive and easily accessible biomarker for the aging process. FaceAge estimations can be obtained by an individual from a simple selfie at home without the need for any specialized equipment. And we can measure a FaceAge at any given moment in the present, or go backwards in time to predict FaceAge on photographs taken throughout an individual's lifetime to look at long-term trends. Additionally, we can obtain repeated measures of FaceAge over short intervals. For example, you can take photographs day to day or hour to hour even. And this serial information might provide interesting quantitative insights into the impact of lifestyle changes or longevity interventions. And lastly, changes in FaceAge and aging rates from baseline in a general healthy population might provide us with early warnings on a change in health status or development of a new disease. So while we started off researching this technology in cancer care, we're really looking forward to interacting and collaborating with researchers in the healthy longevity space as well. So in summary, AI-based quantification of biological age from face photographs provides a non-invasive and easily available biomarker to predict cancer patient survival. FaceAge can improve clinician prediction at the end of life, and we believe that facial health recognition technologies like this can have an enormous impact not only in medicine, but also in the general population, as we now have a simple method to quantify the aging process of an individual and identify people who might be sick just from a simple face photograph. So I'd like to recognize our MGB Artificial Intelligence and Medicine research team, including the director of AIM, Dr. Ertz, who co-led the development of FaceAge with me. Special thanks to Frido and Dr. Lee, talented students who are leading our latest FaceAge research efforts that you heard about today, and also the larger FaceAge team that conducted our earlier work. And I'd also like to thank our collaborators and funding sources, and thank you everyone for your attention, and look forward to the questions and discussion.
Thanks, Ray. It's really exciting and raises a bunch of questions. Before I get to those, though, please, if in the audience, send in your questions. We have room for a number of questions, and we can get to those later in the show. I'm sure there's a lot of thoughts. Um, a lot comes to mind. We've been building clocks in our lab too, and so there's a lot of places we could really take this. One of the thoughts I had is, I guess you have pretty wide range of people that have a chronologic age with cancer. So you can maybe answer this question. I'm wondering how much the variation changes based on the age of the individual. A lot of the clocks constrain that variation by the way the algorithm is written, but we know that the biologic age, or at least I think most of us suspect that the biologic age variation changes with the chronologic age. So I'm wondering if you had a system you could see that in, and if you did see that.
Yeah, that's a really great point and great question. So for the first generation algorithm that I showed you the clinical detailed clinical analyses with, we actually recognizing that variation, potential variation, also recognizing that our cancer population tends to gravitate toward an older age range, like 60 and above. Our first generation model was actually trained specifically on data sets where we oversample for people over the age 60 and older. So that first algorithm is really optimized to perform in a cancer care population, recognizing that exact issue of potential different ranges of chronological to biological age variation. So our second generation model that I described, where we're using that foundation model trained on 40 million images, is trying to overcome that, and we do see that, and we have an additional issue, not just the potential range of variation of biological age from chronological age. We also have in our training data oversampling of younger people, because those are the people that are more commonly photographed. So we had to balance some of that in the training of our second generation model to try to address those differences. But I think, to your point, the biggest problem is that we're still training to what we presume is a healthy population, and that there's such potential inherent variation in the biological versus chronological age, even for a healthy individual, is that how accurate can we get our biological clock, right? And it's going to be a reflection of that underlying training data. So I think ultimately for us, like correlating with eventually validated biological markers may be a direction for us to take, rather than using chronological age as the ground truth.
Yeah, I think one of the things that there's three different clocks that came to mind as you were presenting the data, because one of the questions that would immediately come up is, how does this clock correspond to other clocks?
Yeah,
We've been
exactly.
doing a lot of that work, and I think one of the puzzling aspects of the field is that a lot of the biologic aging clocks don't correspond that much. There is an overlap that's statistically significant, but they're not, as much as you might guess. And I tend to view that as, your different clocks are sampling different pathways controlling aging to some extent. And while there may be overlap, they're weighted in one direction or another. So first of all, I guess before I wanted to bring up three different potential clocks to talk about, but have you compared it to any of the standard methylation clocks or anything so far?
We haven't, and I think that's the key. So I think the comparison part will be really interesting. And then second, with our second generation, the foundation model, we could actually train two biological clocks, right? So instead of saying, take this face and predict what their chronological age would be, take this face and predict use the underlying biological clock to train a new algorithm.
Yeah.
So then you might potentially have a non-invasive readout of the underlying biological clock change over
Yeah.
time.
Yeah.
But yeah, we have not done that correlate. The closest thing we've done so far in terms of access to the data is looking at a lung age, like a physiological lung age based on pulmonary function. And it's interesting that there isn't a great correlation. And I suspect it's because, that you can have, especially in a cancer population, lung damage from smoking that may not be directly correlated with facial changes. So I think it kind of makes sense from an imaging perspective, which is what we have mostly focused on, is that we might have, as I illustrated, like multiple organ clocks, and we're kind of might need to figure out a way to statistically combine those to generate an overall like
Yeah.
physiological biological age.
This raises, I think, a big issue in the field right now that's unresolved is and this is not, cancer is going to confound this. So let's take healthy people to start with. Do you build a top-down system? Like, so recently we built a clinical chemistry clock based on predicting mortality that gives you a biologic age number, and then principal components that
Yeah.
loosely break down into systems, right? So that's a top-down approach. While I mentioned that, it would be interesting. You probably have all the clinical chemistries for some of these individuals that you've done the facial analysis on. So that would be a case where we could compare side by side two different clocks, maybe from existing data. So we maybe talk about that later if you're interested. But that's a top-down approach. Then there's the other approach where some people are more like there's no real systemic aging. It's all different organs doing different things. I'm not really I do think there is a systemic homeostatic process that controls aging, but when you get a cancer situation, it's going to differentially affect some organs relative to others depending on the cancer. I don't think there's an answer to this question right now, but it's certainly what you're highlighting a major question in the field is, do you measure top down or bottom up when you're trying to look at aging?
Yep, absolutely. And I think hopefully where the convergence of the biology and the imaging is that I think there's likely to be a lot of complementary synergy between the two disciplines.
Yeah. We also recently, this is online in bioRxiv, we built a clock from pancreatic cancer patients trying to measure normal aging. And we did it from lipidomics, not because we wanted to scale it, because lipidomics is very hard to scale because of reproducibility from one lab to another. We were more interested in the mechanistic lipids driving aging from that. But it's an interesting parallel because, we built the clock from the cancer patients because we had serum from 500 pancreatic cancer patients at diagnosis. And then we measured it on the normal patients, but we got almost the same data you saw, which is that patients with pancreatic cancer were 8 or 9 years older than normal patients or normal participants using the same algorithm. And we also saw that the biologic age based on that clock was almost as good a predictor of survival from pancreatic cancer as any of the pancreatic cancer markers. So I think that highlights. Go ahead, please.
Yeah, I think that's super interesting that it sort of converges that age differential. And I think one of the key questions we have is, and why we want to look backwards in time if possible, is that it is the chicken or egg thing, right? Are they biologically older because the cancer changed them, or are they at risk for cancer because they're biologically older to begin with?
That's highlighting another big issue, which is that people in our field say aging drives disease. And probably in your field, you say cancer accelerates other aspects of aging. And both of those things are true. So it's kind of a circular process. I would guess that before they have cancer, they're probably biologically older, probably not by the same magnitude. But it's a fascinating question, and we need more perspective analysis to try to sort that out. But also I think this is relevant for biologic age and other diseases as well. So the same sort of analysis could be applied to metabolic disease or potentially cardiovascular disease or certainly dementia. So one of the things that struck me was the low quality photographs that could be used to do this. Because we've worked with, I'm sure you're probably aware, we work with Jackie Han in China, who at Peking University, and we use her facial aging clock, which in our hands is, I think, pretty accurate. But we have to take 3 different images and recreate a 3-dimensional facial construction of the face. So there's a potential for getting more information out of that. On the other hand, it's not as scalable because we need people in the same room under the same lighting, and so there have been attempts to make facial aging clocks from cell phones, but so far I haven't seen one that is super accurate or super. The variation's pretty high in those things, and it's highly dependent on the quality of the photograph too. So I'm really impressed that you got around that with this level of photography, and it would be interesting to compare your clock with Jackie's clocks that have been developed too.
Yeah, I've been following that work, and it'd be super interesting to see it. I think it's likely that the higher the quality of the photo, the more accurate you can get. I think the advantage of our approach is that it unlocks, every photograph ever taken
Yeah,
as a
that
potential
right,
data
exactly.
source.
Scalable.
Yeah.
Yeah. Yeah, so they both may have value, but I think this is anything, I think the reason we build our clinical chemistry clock is it's clinically actionable and scalable because the hospitals collect all this information all the time. So it's kind of the same concept. I really think we need ways to take aging research and get that into the clinic and have it be actionable. And if we do that, that'll dramatically expand the interest in the aging field, as well as maybe provide some insights on what's going on in hospitals. So um, I think it's really interesting what you're doing. You mentioned the ethnicity issue. In Jackie's clock, she had to make separate clocks for African ethnicity versus Chinese Han versus Caucasian.
Yeah.
You have some distribution of ethnicity in your pictures, I'm sure. So, is there a hint how much variation is going to come from ethnicity?
Yeah, there is, because I think in our earlier, our first generation algorithm, where we had access to a less diverse initial training set, when we tried to validate in clinical cohorts, the prognostication seemed to perform not as well. And but with which I didn't show the work here, but our second generation algorithm, which is trained on a much more, larger data set, like 40 million photos, seems to perform better. But I would say for a clinical um, prognostication validation, are the two centers, one from Europe, one from Boston. The patient population is, even though we had, like 20,000 individuals, the percentage that are non-white is relatively small. So we still need further work to identify whether the clinical prognostication is impacted by race. That particularly verifying that it works well enough in non-white populations.
Yeah, yeah, that's there may be some tweaks, that have to be applied for that. But
Absolutely.
I was really, it's nice to see that smoking and excessive alcohol accelerate the age. I was surprised how little substance abuse accelerated aging. I mean, it was
Yeah.
significant, but the magnitude was really small. I guess that substance abuse can mean a lot of different things. So maybe it's just not a very good um, way of identifying what people are actually doing. But you would think
Yeah.
that would be a big driver of aging.
Yeah, and so this was self-reported
Yeah,
in survey intakes from a population study at our center.
yeah,
So we don't have a ton of control over how accurate the data is. There's like a huge category of, like, sort of like maybe, which is
yeah,
not clear
yeah,
what that means. So it
yeah,
could mean things like, recreational marijuana is in there,
yeah,
and maybe that's
yeah,
that's sort of weighing the difference between somebody who's a true, like an IV drug user.
yeah.
So I think that's likely reflected in that, but this is like data that we captured from, like you mentioned, with your clinical labs, like
Yeah.
the opportunistic gathering
Yeah.
of existing data.
There's actually some data too that some drugs like psychedelics may be good for aspects of aging. So
Interesting. Yeah.
it was really fascinating if there was a way to break. I know you don't have that from your data set, but if there's
Yeah.
a way to break this down a little bit deeper, because
Yeah.
um, I hadn't seen much data like that before. I've seen a lot of data on smoking and alcohol, and
Right. Right. Yeah.
um,
No, that's a great point. We can definitely try to dive a little deeper to see what we can find.
yeah. So it's really exciting, Cindy. I think what I'll do is open the questions to Casey, who's here to collate questions from the audience. They're usually better at asking questions than I am, so I'll turn it over to her and let her.
Thank you, Ray, for the very insightful talk and also a very interesting talk. So now I'm just going to give you some of the questions from the audience. So first, it's asked that if understood correctly, a large group of celebrities is being used as a control group. However, celebrities are more likely to have cosmetic procedures to look younger. Therefore, can this group be considered a reliable control group?
That's a great question. So to clarify the point, we trained on that cohort of sort of famous people. And it does include like politicians and not just actors. So, but certainly there could be some plastic surgery impacts within that initial training set. But we validated on sort of non-famous people to show that it works in a healthy population. So that was the first generation, and we certainly recognized that there might be some intrinsic biases in that training set, which is why we came back and did a second generation algorithm that uses a much wider, 40 million face photographs of famous and non-famous people that hopefully captures the full range of facial age. But to go back to the original model, original algorithm, despite that potentially biased underlying training data set, it still works, right? Like it's still a prognostic factor in clinical settings. And that's, I think, our main point, that you can generate, a lot of people can generate facial age estimation algorithms, but we're showing that it's like a clinical prognostic factor. And the plastic surgery thing is interesting, right? Because humans are looking at people's faces and making a judgment on how old or young they appear based on surgeries that have been sort of tailored to satisfy the human aesthetic. But we don't know if the algorithm's looking at the same thing, right? It may not be looking at wrinkle lines on your forehead, right? That slide I showed about the attention mapping was a way to kind of address that question in a different way. What is the algorithm looking at? Temporal muscle, is an area that is, known to be a marker for sarcopenia, right? Muscle loss, muscle wasting. And it's interesting that it tends to that area. So we don't really know if plastic surgery impacts our algorithms, right? And actually, we're looking at a plastic surgery data set now, before and after, to see if our algorithm is impacted by different kinds of plastic surgery, whether it's a facelift or Botox. And certain things like Botox does not seem to alter our algorithm's predictions.
Jackie's picked out the temporal muscles too, as that was a big driver in the prediction, as well as muscles controlling the face.
Yeah, and nasolabial fold was also on our area of tension as well. So it seems like converging to similar areas, and those may not be impacted by plastic surgery. Interestingly.
Okay, so our next question is, can you create a lifetime portfolio from existing photographs to perform meaningful analysis?
Yeah, and that's the data set we're trying to acquire now, like going through a IRB process to see if we can crowdsource photographs over time. Because I think, as Dr. Kennedy mentioned earlier, like there's probably a difference in aging rate once you have the disease. There's probably before the disease diagnosis, whether it's cancer or heart disease, there's probably some sort of accelerated aging, right, process that puts you at risk for that chronic disease. And then before that, the pre-diagnosis, what's changing. And so if we had access to photographs from a given individual, lifetime, from healthy to pre-diagnosis to diagnosis to the chronic disease state, we could really see the impact of aging trajectories on developing disease, and hopefully allow us to backtrack into looking for disease before diagnosis. So that's I think the question's right on. That's where we need to go next. Acquiring those photographs is really just a a human subjects like ethical discussion, but the advantage of our algorithm is it can be run on any photograph of somebody's face that's taken any time in their lifespan, right? And there's trillions of these photographs available around the world, right? So if we can figure out a way to access those, to source those in an ethical way that respects privacy, that respects regulatory concerns, I think will really lead to a more rapid expansion of this facial health recognition field of study.
Along those lines, can you one of the things we can do is de-identify face basically based on those parameters that the computer measures. Is there a way, is that what you're planning to do? Sort of take that, create a set of parameters that generates a face that's analyzable but not identifiable?
Yeah, exactly. So we could extract features and analyze that to get around sort of the privacy concerns. The other way to do it, if we were to collaborate with another center that academic center or that has a large face photograph data set, we've also ported the model to the algorithm to that center, deployed the algorithm at a given center. So the data, because I think you're alluding to the fact that in the medical setting, face photographs are protected health information, right? So we can't share those as easily across centers. So we sort of ported the model to our collaborators to address that issue.
So I do have a question of my own. I'm just wondering, do you review your FaceAge result to patients, and how do they usually react if they know that they are biologically older, especially that it's associated with a prognosis?
Yeah, we have not shared that yet. So that would be the next phase of this research in cancer care, would be trying to implement it into the clinic. I think for us, we'd be mainly focused on that sort of end of life situation where we're trying to help clinicians make a better decision on how aggressive to be with care. It's a great question of how patients would feel in that situation if they're told that they look much older. And I think it's a matter of framing, right? It's not just the age part. It's about we're concerned that this biomarker is saying that you're getting closer to the end of life, and we have to have a serious conversation about what kind of cancer care is appropriate.
Yeah.
We're having those kinds of conversations every day as oncologists, but instead of having a quantitative measure, we're using our subjective clinical intuition, right? You don't look good. I think you might be, the end is coming soon. And so our intent, our original motivation was really just to develop a tool to help quantify, standardize that clinical intuition.
I think, and it's probably going to be different in palliative care, but I can already tell you how people react in the healthy population, which is that if you tell them they're younger, they're very happy. And if you tell them they're older, they tell you your clock doesn't work.
Yes. I would suspect the same.
So my next question is, has there been cases where prediction of FaceAge, the result is younger than the chronological age? And especially in cancer patient, if that occurs, what sort of conclusion can you make out of this?
Yeah, I didn't emphasize that as much, but people that were much younger than their chronological age by appearance, particularly around, our cutoff is around 5 years. So if you're 5 years younger than your chronological age, those people did the best. They had the best outcomes across multiple cancer types. So how you might use that, I treat lung cancer. A lot of the treatments that we offer for lung cancer are quite toxic. And so, 80, 90-year-old that looks their age and acts their age, sometimes we have a frank conversation that the treatment may be something that's more harmful than curing you of the cancer, that you may actually die from the treatment, just given your health status. But we have 80 or 90-year-olds that look like 60. And we've run the model on some of these people, and the algorithm concurs that they look like a 6-year-old. They're still snow blowing, shoveling snow at age 80 and 90. And those people, the clinical intuition is you treat them that way, right? That you treat them as if they're a 60-year-old, and that means aggressive cancer treatment. And so that's how it alters our care. And I think having, it's my intuition at this point, right? But having something like FaceAge would back that intuition, right? To say like this guy does look like a 60-year-old, or other biological age estimates, right? To support that this is a person that could tolerate more aggressive treatment.
You might make the assumption, and maybe you don't have enough data to assess this, that a cancer like lung cancer, especially where it's driven by smoking,
Yeah.
that might, escape the biologic age, assessment more than cancers like prostate cancer that are heavily linked to aging. And so I wonder if you can look at that, where cancers that have clear risk factors that are very high risk factors, if that the biologic age is maybe less, maybe I'm not going to say it's less meaningful. It could still be very meaningful, but it may be less accurate in predicting who's getting the cancer.
Yeah, that's a great question. We haven't looked at that, but that's certainly something we could, yeah.
In what specific clinical scenarios beyond cancer prognostication could FaceAge be applied to improve patient outcomes or support medical decision making? So beyond cancer prognosis.
Yeah, I think any situation in medicine where you're factoring in somebody's chronological age at this point, which is basically, many aspects of medicine, right? Like surgeons are trying to make a decision whether they should do a hip replacement on somebody who's 80. But what if they look like a 60-year-old? Then that's so I think, as doctors, both in existing clinical decision support tools where we put in a bunch of clinical factors and make a treatment decision, chronological age is already a part of many of those calculators. And then layering that with that clinical intuition, like the qualitative assessments that doctors are making every day, we really think that this could be a tool in any aspect of medicine. And we're still thinking through like sort of where are the most impactful places we could clinically deploy this. And I think it's basically where people are using age as a big driver of their decision making, like who gets a transplant, those kinds of things. And so in some, I think anywhere in medicine.
Okay, so I think we just have time for one last question, maybe. Do you think your model can predict cancer incident from younger population? So we know that right now a lot of cancer going down the younger age than before. So what do you think?
Yeah, so we're moving from then a face age to a face diagnosis, right? And that's why I use the term facial health recognition at the beginning of the talk, is because I think there's a possibility that we can get to that point, develop the technology that we can start predicting risk. I won't say like, predicting the cancer. I'll say risk, predicting cancer risk or predicting chronic disease risk based on face photographs. So I think that remains to be proven. I think the technologies are there now in terms of the underlying AI technology that describe the foundation model for us to test that hypothesis. We just need the right data sets now, which is, taking a patient at diagnosis and then getting photographs tracking back throughout their lifespan. That's the data set we're trying to acquire, and that will help answer that question whether we can make a face diagnosis.
I wonder whether you could also add some other measure that's relatively non-invasive, thinking like a strength measure or pulse rate velocity or something that is relatively easily done, and then couple that so you have two parameters to look at, and that may give you a more meaningful prediction.
That's a great suggestion. We have a prospective trial that's in IRB review, ethics board review, where we're going to pair FaceAge with a get up and go test, prospective collection, so just to sort of see how well it correlates with the functional measure.
Yeah, that's great. Well, thanks, Casey, and thanks, Ray. We'll come back and get an update at some point. It's really interesting data. Looking forward to hearing what comes next.
Yeah, thanks for the invite. Really enjoyed the discussion.
Anytime. I want to remind participants to use the chat function and panelists and all attendees option to leave comments and feedback. We always want to know what you're thinking and how we can find new topics for new shows. Our next Healthy Longevity webinar will be March 20th with Professor Nazif Alik from UCL, University College London. And he hasn't given us a topic yet, but I'm going to bet that it's interesting. And finally, we'd like to leave you with a thought-provoking and uplifting video titled How to Embrace Aging and Overcome the Fear of Growing Older, Curler Confessions. It's a wonderful exploration of finding joy and wisdom in the journey of life. So enjoy the video and come back next week. Thanks, everyone.
Now for Curler Confessions, where we get real about some real issues. Okay. Carolyn, this has me asking you, how do you feel about sharing your age with people? Don't be ashamed of being 23.
I love you. Are we really? You guys, if you know me, you would never have been ask me that, because we know the answer. I don't think any woman, I'm a vampire, I do not share my age, and it's none of your business, okay?
That's right. So in this morning's Curler Confession, with that said, Charlotte Today host and life coach Colleen Odegaard talks about why she thinks we should embrace.
I'm happy she thinks that.
Getting older.
I confess I just turned 49 years old, and I have no problem sharing my age with people, and it's because I refuse to feel ashamed about getting older. Culture would lead us to believe that we are less attractive as we age, make us feel bad that we don't look the way that we used to, but I reject it. I just don't believe it. Instead, I sink into gratitude and self-acceptance. That's the antidote. I focus instead on how grateful I am to be alive, to feel my feet on the planet Earth. I'm thankful for my body, even the saggy parts. I'm grateful for the wisdom I have now that I didn't have when I was younger. I make a decision not to beat myself up, but instead to talk to myself like I'm a dear friend. So many of us spend so much time feeling bad about ourselves because of the chatter in our minds. I'm too fat, I'm too old. But instead, sink into gratitude. That is what I choose, and I want that for you too. Bonus confession, I could use a little Botox right now, but even if I don't get it, I'm still grateful.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.